So nitrogen fixing bacteria. So bacteria are very helpful to fixation of the nitrogen from atmosphere. The bacteria are maybe the in symbiotic manner or maybe asymbiotic manner. They are fix the nitrogen from the atmosphere due to the presence of nitrogenase enzyme, which involves dinitrogen or molecular nitrogen is converted into the ammonia, which is usable to the all higher plants. So the symbiotic bacteria and free living bacteria, along with these bacteria, the blue-green members also are cyanophyceae members or cyanophyceae algae, cyanobacteria members like Anabina, Nostoc. These algae members also, blue-green algae members also involve the fixation of the nitrogen from atmosphere due to presence of heterocysts. These are the specialized cells which are available, which are present in the trichomes of the blue-green algae members. Then some of the nitrogen fixation bacteria along with the cyanobacteria members or other nitrogen fixing members. I will show the tabular form. So this slide shows about the different categories of the microbes which are involvement of the fixation nitrogen from the atmosphere. The first one, the nitrogen fixing bacteria in symbiotic manner. The symbiotic biological nitrogen fixation. The bacteria are the mutualistic relationship with plant roots and fix atmospheric nitrogen. So rhizosphere roots are the higher plants roots are associated with fungal members that we can call as mycorrhiza. So in this mycorrhiza is also one of the most suitable for the fixation of the nitrogen. But here the fixing nitrogen bacteria, the best example of this Rhizobium leguminosarum and other Frankia, Rhizobium leguminosarum, which induces the root nodules in legumes, whereas Frankia is also another type of bacteria which non-leguminous, which induces the root nodules in other leguminous plants. These root nodules are the suitable area or suitable place fixation of the nitrogen from atmosphere. In these root nodules contains the nitrogenase enzyme, which is helpful for the fixation of the nitrogen. The best examples of this the symbiotic nitrogen fixation of the bacteria Rhizobium and other Frankia. Here the improvement of the nitrogen availability in soil reduces fertilizers and dependency and enhance crop growth we are observed through this symbiotic nitrogen fixation. And nitrogen fixing bacteria, the free living bacteria. The free living bacteria, the best examples of the free living bacteria, that is Azotobacter, Beizeringia, and Clostridium. These are the bacteria which they are independently fixation of the nitrogen from atmosphere. They are not associated with any other plants. They are lived in a free manner in normal atmosphere. But these microorganisms are fixing the nitrogen from atmosphere, which enhances the soil fertility in non-leguminous crop plants and promote plant growth also. Then nitrogen fixing cyanobacteria. We know that some of the blue-green algae members like Anabina nostoc, these are helpful for the fixation of nitrogen in the heterocyst cells, which improves the nitrogen levels in paddy fields, especially in nowadays the paddy crop is highly increasing the area, like quantity and also the quality also. Why? Because the availability of the water and also the seasonal supporting, but due to the heavy rainfalls, though the increasing area of the cultivated croplands like paddy. So in these paddy fields, this Anabina nostoc is very useful for fixation of the nitrogen in symbiotic manner through by using azolla cultures. Azolla is a water fern. It is an aquatic fern. Azolla leaves. Azolla, it is a belongs to fern plants, pteridophyta plants. So this plant is associated with Anabina. Anabina azolle is the best example for the nitrogen fixation in the paddy field. So when we are using these azolla cultures in paddy field, the increase of the nitrogen and also useful for the crop improvement. But farmers they are not able to, or even though they are not supposed to use more quantity of the chemical fertilizers. And phosphate solubilizing mycorrhiza, microbes, PSM. These PSM organisms are converts insoluble phosphorus into soluble phosphorus. Why? Because whenever these dissolved in water or soluble conditions, then only plant roots or the root hairs are absorbs these nutrients from the soil. So that's why the phosphate solubilizing microbes like Bacillus and Pseudomonas aspergillus, these are the microbes helpful for insoluble phosphorus into soluble phosphorus, which improves the enhance the phosphorus uptake and promote root development, and also improves overall crop productivity. And potassium solubilizing bacteria, KSB. In previous, what we in before we are what we discussed the PSM, phosphate solubilizing microbe, along with this potassium solubilizing bacteria. Why? Because the major nutrients, the macronutrients, are very important for growth and development of the plants, including the fruit yielding and also the flowering. So that's why the all metabolic activities or physiological functions are without any interruption. These minerals are necessary, are essential. So due to that reason, the microbes also very helpful to the crop improvement and also plant growth. Very helpful to the crop plants. And the potassium solubilizing bacteria examples are the Fruitaria, Aurantia, and Bacillus mucilaginous. These are the microorganisms which are very suitable for the soluble potassium from minerals into plant available forms, which improves or which increases potassium availability, enhances stress tolerance, and boost crop yield. We know that some of the biotic stresses and abiotic stresses, we know that. So abiotic stresses are water stress, drought stress, cold stress, chilling stress, temperature stress. So these type of stress conditions, this potassium is helpful to the tolerant to the different types of the stress conditions. And plant growth promoting rhizobacteria, that is PGPR, plant growth promoting rhizobia. These are also enhance the plant growth by producing growth hormones and suppressing pathogens. Right. So the Pseudomonas fluorescens and Bacillus subtilis, these are the microorganisms or the bacteria or rhizobacteria improves root development, and also along with root development, disease resistance also increase the resistance to the different pathogens of the microbes, whereas bacteria, fungi, and viruses. Okay, then mycorrhizal fungi. Already we discussed in the previous slides, ectomycorrhiza, endomycorrhiza. Okay, these are mycorrhizal members. Examples: Glomus rhizophagus increases nutrient uptake, phosphorus, zinc, enhances drought resistance, and improves soil health. So microbes in disease resistance. Disease resistance. What is the definition of this disease resistance in the biocontrol agents of the against pathogens? The biocontrol agents are beneficial microorganisms in bacteria, fungal members, viruses, and also nematodes that suppresses the plant pathogens. They provide an eco-friendly alternative to chemical pesticides. And mechanism of the biocontrol. Which type of mechanisms adapted to the control of the other pathogens by using of these microorganisms? Number one, competition, which is very healthier practices in biocontrol mechanism. The biocontrol agents compete with pathogens for space and nutrients. Like the best example of this competition, that is Pseudomonas species. These are the bacteria. And some of the second mechanism of the biocontrol is antibiosis. Antibiosis. Which this production of antimicrobial compounds that inhibit pathogens. The best examples of this microorganism, that is Bacillus species. Then parasitism. Parasitism is another mechanism of the biocontrol which direct attack and destruction of the pathogen. The best examples of this parasitism, trichoderma. It is a fungal member which control the other fungal members to development of the root growth and also the crop improvement. And also the other bacteria also. Some of the bacteria, Delovibrio bacteriovorus. Delovibrio bacteriovorus is another type of microorganism. It is a bacteria which kills the other bacteria. It is a predator bacteria which uses the cleaning of the sacred river Ganga. గంగా నదిని పరిరక్షించడానికి, గంగా నదిని పరిశుభ్రం చేయడానికి, గంగా నదిలో ఉన్నటువంటి కలుషితాన్ని నాశనం చేయడానికి, కలుషితాలను నివారించడానికి డెలోవిబ్రియో బ్యాక్టీరియోవోరస్ అనేటువంటి బ్యాక్టీరియాను వినియోగిస్తున్నారు. డెలోవిబ్రియో బ్యాక్టీరియోవోరస్ is another predator bacteria which suppresses other bacteria, harmful bacteria for the cleaning of the sacred river Ganga. మనకి ఇప్పుడు ఆల్రెడీ ప్రయాగరాజ్లో ఏమొచ్చింది, 144 సంవత్సరాల ఏమొచ్చింది, మహాకుంభమేళ వచ్చింది. మనకు తెలుసు ఈ టైంలో మనకు ఎక్కువ మొత్తంలో ఈ మన మైక్రో ఆర్గానిజమ్స్, పెస్ట్, ప్యాథోజన్స్ అనేటువంటి మనకు అటాక్ చేసే ప్రమాదాలు కూడా ఉన్నాయి. వి నో దట్. దెన్ ఇండ్యూస్ సిస్టమేటిక్ రెసిస్టెన్స్, ISR. In this mechanism, activating the plants by themselves, immune response against pathogens. వాటికవే ఇమ్యూనిటీ పవర్ను పెంచుకునేటువంటి విధానం అన్నమాట. నెక్స్ట్, common biocontrol agents. Nowadays, we know that some of the other common biocontrol agents we are observed in the commercial aspects. In bacteria, biocontrol agents are Pseudomonas fluorescens, Bacillus subtilis, and Streptomyces. These are the bacterial organisms or bacterial microorganisms which are commonly used for biocontrol agents from different pathogens for crop improvements, for sustainable agriculture, and also then the eco-friendly manner. Then fungal members. The common biocontrol agents of the fungal members are Trichoderma viride. The high amount of the fungal member usable, very vastly, widely used fungal member of the biocontrol agents are Trichodermas. Then Beauveria, Bassiana, and Gliocladium. Some of the viruses also used as a biocontrol agents from the pests and pathogens for protecting the crop plants and improvement of the crop. They are the baculoviruses targeting insect pests. These baculoviruses are targeting the insect pests on the crop plants. And some of the nematodes also used as a biocontrol agents for the control of the pests and other harmful microorganisms to the crop plants. They are Stenionema and the Heterorhabditis. These are two nematodes, nematohelminthes members or organisms which are useful for the biocontrol agents for the crop improvements. And some of the benefits of the biocontrol agents. What are the beneficial control agents? Number one, environmentally friendly and sustainable environment. These are also used for reduces dependency on chemical pesticides. Why? Because these are all naturally used as a biopest and also the biocontrol agents. So that's why they are less depends or reduces the dependence on chemical pests, pesticides, which is also used as enhances soil and plant health. Soil health is important. By the by, plant health is also important. Soil health important. At the same time, plant health also important. Okay. Then low risk of pathogen resistance. Then next one, the application in agriculture. Number one, seed treatment. Seed treatment is one of the most important practices for the all farmers, whether they are marginal farmers or the large scale farmers. Definitely, we suggest them to, before going to sowing the seeds, should practice this seed treatment. It is a very easiest practice, protecting from the other plant pathogens, other pests. The seed treatment with trichoderma. Trichoderma is a fungal member which is very helpful for the seed germination. Seed germination is promoting by these trichoderma species. Okay, and also protect from the external environment. And soil amendment to control root disease. Some of the plants, very rigorously, they are going to wilt by decaying of the root system or by infecting of the other pests or other pathogens in the area of the rhizosphere or root pot. So that's why the soil amendment to control root disease by using of these biocontrol agents. And foliar sprays to prevent fungal infections. Along with this, used integrated pest management programs. And induced systemic resistance. Induced systemic resistance is a plant defense mechanism acted by beneficial microbes, helping plant resist to wide range of pathogens. The mechanism of this induced systemic resistance is, number one, microbial recognition of the beneficial microbes like Pseudomonas, Bacillus. These are trigger ISR in plants. ISR means induced systemic resistance. And what are the signaling pathways for this induced systemic resistance? That is jasmonic acid. Jasmonic acid and ethylene pathways. These are the two pathways are the key hormones involved in ISR that induce systemic resistance. Jasmonic acid is a phytohormone along with ethylene. And these are, along with these, defense activation by strengthening of cell walls, enhance enzyme production, and increase secondary metabolites. Some of the microbes involved in ISR, like Pseudomonas fluorescens, Bacillus subtilis, and Trichoderma species. These are the microorganisms involved in induced systemic resistance. And what are the benefits of this induced system resistance? Protects plants against fungal, bacterial, and viral pathogens. Environmentally friendly or eco-friendly for sustainable environment, and enhanced plant growth and stress tolerance. Stress tolerance, different biotic and abiotic stresses, we know that. Okay. And antimicrobial metabolites from microbes. Some of the secondary metabolites, they are used as antimicrobial metabolites, which are released or produced from microbes, like some of the bacteria and other microorganisms. Antibiotics like streptomycin, which is synthesized or produced from streptomyces bacteria, and bacitracin. Bacitracin is another type of antibiotics. It is a broad spectrum antibiotic which is synthesized by Bacillus subtilis. These are the two bacteria which involve for the synthesizing antibiotics. And lytic enzymes. Lytic enzymes like chitinases, proteases, glucanages that degrade fungal cell walls. And some of the toxins, the best examples are iturin from Bacillus and trichothecins. These are from Trichoderma. And volatile organic compounds inhibit pathogen growth and trigger ISR. Some of the applications of these applications in agriculture, these are used in biopesticides and biofertilizers, and also reduce the use of chemical fungicides, and improve soil and plant health. What we are observed from these important points, we know that we are knowingly or unknowingly used to intake the food material, lots and lots of harmful chemical substances. So that's why our ultimate goal is reduces the chemical fertilizers and chemical pests and insecticides for better health and also sustainable agriculture, but at the same time sustainable environment. Are you able to follow my class, my dear friends? Okay. Then what are the biotechnology approaches in microbial applications? This is the last topic in our today's national webinar. Number one, the biotechnology approaches in microbial applications. Number one, agriculture, that is biofertilizers. We know that very well-known biofertilizer is Rhizobium, Leguminosarum, Azotobacter. These are the nitrogen fixers. And biopesticides like Bacillus thuringiensis, BT bacteria. Bacillus thuringiensis is a natural insect control or bioinsecticide. We can also call it as bioinsecticide. These BT or the biopesticides contains the crystal genes which are helpful for the control of the insecticides or insecticide larvae, which are depends on cotton bollworms. Then some of the other the medicinal applications of the microbial biotechnology. Number one, antibiotic productions. We know that antibiotics are the first antibiotic. We know that the wonder drug we are well known, that is penicillin. It is discovered by Alexander Fleming in very ancient, the first and primitive and foremost antibiotic. But nowadays, so many antibiotics are produced by using these biotechnological approaches in genetic engineering process, by using genetic engineering approaches. In this regard, some of the, not only the penicillium, but also other bacterial members also used for production of the antibiotics in large scale, in commercial aspects. Okay. Then the vaccines. Very, very important term, vaccines. In 2020 onwards, we are facing very pandemic situation. We are recovering that situation. Everybody has suffering. Everybody has conscious about the health. At the same time, awareness about the vaccines. But our sacred nation, our India, has produced the vaccine from corona, for corona, preventing corona. So this vaccine is produced by involvement of these biotechnology approaches, genetic engineering approaches. Some of the industrial applications, fermented food. Whenever our civilization has occurred, from ancient to modern man, fermentation is one of the best and foremost suitable biotechnological approaches in commercial aspect, in industry, beverages industry, and also alcohol industry and bakery industry, they are depends for fermentation process for food industries also. The best examples of this microorganism, we know that Saccharomyces cerevisiae. This is, we can also call as baker's yeast and brewer's yeast. Okay. And which is very extensively used in all. మనం పొద్దున్న లేవగానే ఏమంటాం, మనోడు బేకరీకి వెళ్దామంటారు. ఆ బేకరీ అంతా కూడా దేని మీద ఆధారపడ్డదంటే మిత్రమా, మన సాకరోమైసిస్ సెర్వీసియా. దీన్ని మనం ఈస్ట్ అని మనం పిలుస్తాం. ఈస్ట్ ఇస్ ఎ సింగిల్ సెల్యులర్ ఆర్గానిజం విచ్ ఇస్ బిలాంగ్స్ టు ది ascomycotina fungal member. And enzyme production. Enzymes also very, very important in industrial sector for production of the preservatives and also health aspects. So the microorganisms are involved for production of these enzymes are aspergillus species for amylase in detergents. If we are using so many different types of detergents, in the detergents, the aspergillus are one of the suitable microorganisms for the enzyme, that is amylase. And environmental biotechnology. Without environmental conscious, we are unable to survive and sustain for better tomorrow. So for better tomorrow, we are definitely conscious about the harmonious environment and sustainable environment. In this regard, biotechnological approaches or biotechnological applications are most suitable for the environmental protection. Number one, bioremediation. In this bioremediation, Pseudomonas is a bacteria which is discovered by Anando Chakravarti, our Indian scientist, Indian professor, Acharya, Professor Anand Chakravarti, who discovered the Pseudomonas putida. This bacteria is useful for the removal of the oil spills from the where the oil ores are. మనం సముద్రం పైన పెద్ద పెద్ద నావలు వెళ్తా ఉంటాయి కదా, మన బిగ్ షిప్స్. సో దీస్ షిప్స్ ఆర్ due to climatic conditions, they are sink in the oceanic conditions. So in that way, in that region, the oils are cover the surface of the oceanic environment. In that condition, the Pseudomonas putida is best suitable for removal of the oil spills from that environment. And wastewater treatment, the rapid urbanization process, the industrial sector, industrialization process. Our era is also the globalization era. Urbanization process is rigorously developing. In that way, the solid waste management is one of the best, the most important challenge to the all governments. In this regard, the biotechnological approaches to removal of or the treatment of these wastewater and also the solid waste by using of this microbial organisms. Am I audible? ఎవరు మాట్లాడట్లేదు. Then genetic engineering of beneficial microbes. So in these techniques, the genetic engineering of microbes, number one, the recombinant DNA technology or DNA technology or chimeric DNA technology. In this technology, we are using or we are inside the gene which are beneficial gene or usable genes. Insertion of the beneficial genes by using of the vectors or by using of other microorganisms like the Escherichia coli for production of the human insulin. So many people, they are suffering from the knowingly or unknowingly suffering from diabetes. So type 1, type 2 diabetes, we know that. So during this situation, our Escherichia coli is producing high amount of the insulin by using of these biotechnological approaches. Then CRISPR-Cas9 technology. It is very most important technology for gene editing. Gene editing is one of the most important advanced technology for the editing the genome from strain improvement. And synthetic biology. This is also very important for production of the biofuels. Biofuels is remedy for or substitute for what we can say that fossil fuels. Okay, and BT crops, the genetically engineered or genetically modified organisms. That is number one, BT crops or BT cotton. Bacillus thuringiensis contains the crygenes Cry1 AC and Cry2 AB. These are the genes which are held responsible for the suppressing the larvae of the different insects on the cotton plants, especially BT cotton. This is the best bio pest used as a bio pest. And GM probiotics, the engineered bacteria for gut health benefits. GM probiotics are used, and oil degrading microbes. The Alcanivarax is the strains enhanced for bioremediation for using the oil degradation from the external environment. Now CRISPR-Cas genome editing microorganisms will discuss in this slide very within 1 minute. So this is the clustered regulatory, the interspaced short palindromic repeats, that is CRISPR-Cas, is a revolutionary genome editing tool used to modify microbial DNA for various applications. The applications of the microbial biotechnology of this CRISPR-Cas, that is agriculture, for enhancing nitrogen fixing bacteria for better crop productivity. In medicine field, engineering bacteria to produce antibiotics and vaccines. In industry, improving fermentation microbes for biofuel and engine production. In bioremediation, developing microbes that degrade pollutants more efficiently. And development of the biofertilizers and pesticides. We know that biofertilizers, Rhizobium, Azospirillum, and biopesticides, we know that Bacillus thuringiensis and Trichoderma. Bacillus thuringiensis from bacteria and Trichoderma viridae from fungal members, and Pseudomonas fluorescens from bacteria. These are all the common biopesticides useful for the crop improvement and also used in sustainable agriculture. And some of the case studies I am showing here for the recent advances for the microbial biotechnology. Number one, CRISPR edited microbes for sustainable agriculture. Number one, Rhizobium and Azospirillum bacteria using this CRISPR technology to enhance nitrogen fixation. And microbial enzymes in industrial biotechnology, Bacillus subtilis and Aspergillus niger. These are the microorganisms from bacteria and fungal members used for large-scale enzyme production. The best example, the enzymes are amylases and proteases. And results of these microbial enzymes in industrial biotechnology, more efficient and cost-effective process in food and textile industries. And biofuel industries, engineered Escherichia coli and yeast. These are used for production of the biofuels and biodiesel production. We know that biodiesel is the production from the, we are using the, మనం చెప్పుకుంటాం ఈ అడవి ఆముదానికి మనం జట్రోపా is the one of the best suitable for the large scale cultivation by promoting. The government has also given the subsidies to cultivate the biodiesel plants like Jatropha in 100% subsidy. We know that. And some of the microbes in bioremediation, Pseudomonas putida and Deinococcus radiodurans, used to clean up oil spills, heavy metals and radioactive waste. Pseudomonas putida discovered by Anand Chakravarti. We know that. And result that significant environmental restoration in polluted areas. So oil spills are dumped on the surface of the oceanic environment, that is removed by this Pseudomonas putida. And some of the success stories, microbial biotechnology, by using this BT cotton and the breakthrough in pest resistance. So Bacillus thuringiensis, whenever we are using this BT cotton, the reduced pesticide uses and a high yield increase for the farmers. And probiotics for human health. Lactobacillus, and bifidobacterium. These are the two strains used for production of the probiotic supplements and dairy products. And COVID-19 vaccines. We know that mRNA vaccines, that production of Pfizer and Moderna, the leveraged microbial fermentation in Escherichia coli for vaccine component. The result of this vaccine, COVID vaccine development and effective response to the global pandemic situation. Our India has sent to the so many countries for protecting the value life of the human health condition. And microbial plastics and biodegradable solutions. The Pseudomonas and Ralstonia, these are the engineered to produce bioplastics. The result, reduction in plastic pollution and sustainability in packing industries. And conclusion, enhanced agricultural productivity, environmental protection, medicinal advancement, industrial innovations, and cost-effective and sustainable agriculture and sustainable crop improvement. We are get benefited by using of this microorganisms through biotechnology, and call for sustainable agricultural practices, encouraging bio-based inputs, reduce the chemical dependency, adopt climate resilient strategies, strengthen research and innovations, policy and public awarenesses. These are all for call for sustainable agriculture. So, my dear friends, thank you very much. Regarding of this webinar, if you have any doubts, please you may ask. I will clarify your doubts. Anybody else? Okay. Anybody else? Okay, my dear friends, thank you very much for your patience.